Hey guys, we are back for week 5. This week we are talking about some basic cell biology. I'll try to make it somewhat painless. Genetic regulation of cellular function, because that's important to understand. And then lastly, we'll get to epigenomics. You probably won't see that for another day or two. But we'll start here. Most of it's review. There's a nice picture, pretty cell. Basic review of cell biology. You can kind of go through this really quickly. If you remember anything from 10th grade or from your gen eds or your prereqs for nursing, we'll just kind of do an overview of the cell. So here's a generic cell. A mature cell has its own appearance. It matches its unique function. Skin cells might make keratin or collagen. Glandular cells produce and secrete hormones. Skeletal muscle cells contract to help the body move. Bone cells provide structure, on and on. They all look different. This is sort of the generic version of a cell. Most mature cells have the nucleus, and we know the nucleus is where the chromosomes are. That was a really good test question. A few chromosomes, and they're circular in shape. They're very simple chromosomes, so found here in the mitochondria. We'll get more into that when we talk about the sex-linked disorders and sex-associated disorders. There's a lot of cytoplasm in relation to the nucleus. When it's not in mitosis, the nucleus tends to be very small. The mitochondria is where we have energy production in the form of ATP. There is a small amount of DNA, just 37 genes as compared to the 25,000 we see in the nucleus. Now that was the generic cell. Here's some examples of what mature differentiated cells look like. So here's a neuron here where my cursor is circling. Here are some osteocytes, bone cells. You can see they look very different. They all have a nucleus, all these different types. This is the smooth muscle of the intestine here. And you can see the elongated sort of shape. The nucleus is sort of flat and long. So each cell has its own appearance that matches its function. Its structure matches its function. And even though each one of these cells has the same 46 chromosomes, all normal, there are genes that tell the cell which proteins to make, what structure to assume, like what form it's going to take, how long it's going to live, when it reproduces, if it reproduces. So that is all regulated by genes. And we're going to find out how that is. So cells are held together by cell adhesion molecules, these little fibronectin's an example, integrin, these little red dots here sort of represent the glue that holds the cell together. And cell adhesion is important because when cells aren't adherent to other cells, there are signals that come into that cell and tell it to reproduce. When the cells are touching each other, they know. It's like, okay, stop. There's enough of us now. If they lose that ability to suppress reproduction, then that's when they become a problem we call cancer. Anyway, cell growth happens in a couple different ways. There's contact inhibition. That keeps the cells from multiplying. When cells are in close contact with one another, they are inhibited from mitosis. That's one of those things that tells the suppressor genes, keep everybody in line. If you have a loss of contact with those other cells, let's say you get a cut, or there's some other loss of healthy cell tissue, if a cell is able to undergo mitosis, it's going to get a message that's going to tell it it's time to make more cells. Some cells never undergo mitosis once they're mature. Examples of that would be brain cells, nervous tissue, cardiac muscle, skeletal muscle. Those cells do not reproduce. So if you lose them, they are replaced with scar tissue, and all that scar tissue will do, it will not do any of the function of those cells. It just kind of keeps them from leaking. It keeps the structure intact. But that's why, heart damage can be so dangerous. You have a couple of cardiac events, minor or major. You have scar tissue in the heart. Eventually, you're going to end up with congestive heart failure. You don't have enough contractile tissue. It's been replaced with scar tissue. So it's kind of important to remember. Cell growth is governed by two things. Oncogenes, they're like the director that yells action. And suppressor genes, They're the genes that yell cut, if you want to think of it that way. Other people like to think of the oncogene as an accelerator and a suppressor gene that is like a brake. So one says stop and one says go. Cells that can go through mitosis, so your skin cells, liver cells, the cells of your intestinal lining, those kind of cells undergo mitosis and they make more cells. And that process is called hyperplasia. They grow more cells. Okay. Plasm or plastic, those are sort of the root words that tell you that something is being manufactured. Oncogenes are going to signal those kinds of cells to undergo mitosis and make new daughter cells. Cells that don't go through mitosis, those brain cells, mature muscle cells, heart cells, the cells will get larger, but they will not produce more. That's hypertrophy. So that's when you see, if you ever see an echo report, somebody who's had heart disease for a long time, they have hypertrophy. They have enlarged, they'll have an enlarged heart, but the cells themselves are actually enlarged as well. So they get bigger. Or skeletal muscle. When you train and your muscles get bigger, they're not growing new muscle cells. The cells themselves are getting bigger. So those are sort of important concepts to remember. We're going to go into mitosis a little bit. I don't need you to have that picture in your head, and I don't want people going crazy with anaphase, telophase, metaphase. I want you to understand how the overall process works and how it's governed by genes. This is a course in genomics. It's not a course in cell biology, even though we have to address it to some extent. Most cells live their lives in G0, okay, right here. They're busy performing the functions of their cell. If they're, a cell that produces mucus, that's what they're doing. Excuse me. If they're a cell that secretes a hormone or an enzyme, that's what they're doing. They're living their lives as cells. They live in G0. And the suppressor genes make sure that they stay at their jobs, at their posts. However, when there's a need for new cell growth, that cell can be prompted to go into G1, which is the first phase of mitosis, by the oncogenes. Suppressor genes are silenced, okay, for that particular cell. So here, this is just a brief overview of the cycle. G1, everything gets multiplied. The chromosomes get duplicated. It takes about 8 to 12 hours. So you just have like copies of each chromosome. And then they're replicated in the S phase. Then protein synthesis, you're going to make your histone proteins and other things that the cell needs. The, Golgi body separates, but you're going to make ribosomes, all the other cell structures. And at this point, genes that are programmed to check for mistakes in the code are going to look. They're going to scan and look for errors. And if anything is found, it's going to sort of put a halt to it. And until you can put them back together, no further progress happens. Okay. So you go through all these checkpoints to make sure that all the cell division is appropriate and that the cell is healthy enough to withstand the process. That's the first checkpoint. And then once it starts, there are these internal signals, and that's all gene regulated. There are genes that actually control this. There are chemical messengers called cyclins. I don't need you to memorize this. Cyclin-dependent kinases, tyrosine kinase, and they kind of keep it going. Each one will say, okay, now this, now this. They're sort of like little teleprompters telling the cell what to do. So those checkpoints exist to scan for errors, like I said, and the genes regulate whether the cell is ready to progress to the next phase. Usually when that cell gets arrested in the cycle, instead of trying to continue with the mistakes, the cell will die. If there are no errors, eventually you're going to go through those other phases, prophase, metaphase, anaphase, telophase, all going to be packaged up very nicely, where you have 46 chromosomes in each cell, wrapped up with their little centromere. everything is in order, and you have 2 new daughter cells that result from the duplication and division of DNA. If this is super complicated, this is where you want to go to your book. I can't promise they'll make it simpler because they kind of don't, but just know it's hard to get into mitosis. Once you're in, you have to listen carefully to the instructions and several points. There's quality control. Okay, that's what I kind of want you to come away with, that there's quality control to this process. It is not a free-for-all. We don't always want to be growing and reproducing cells. And when we do reproduce cells, we want it done precisely right. Okay, that's what you need to know. Apoptosis. This is a really super important concept for you to grasp. Apoptosis is a programmed cell death. After a genetically programmed number of cell divisions, the cell is programmed to die. Okay? That number can be altered by a few things, but keep it simple. After it's been on a number of cell divisions, it's retired. It goes through a death. It's been replaced by enough cells so the tissue and organ maintains integrity. It's modulated to some extent by those telomeres. Aha, there's a reason that I incorporated that into week 2's lecture. Telomeres, those little caps, like the shoelace caps. I did find the name for them, by the way. It's called an aglet. Those caps keep the chromosomes from unraveling. And the telomeres are maintained by an enzyme called telomerase. And that maintains the telomere length. Depletion of telomerase leads to unraveling of the chromosomes, which starts a whole cascade of things that you do not need to memorize. But basically, when the cell gets old and the chromosomes are unraveling, there are these chemical signals that start a cascade. Eventually, the cell sort of breaks down. It gets smaller, it has these blebs, but eventually it breaks down and then macrophages come along and eat them. And that is a normal, healthy process. We want apoptosis. We want to replace damaged cells, older cells. It ensures that the cells that are left behind in that tissue or organ are functional, that they are doing their best. Any cell that relies on mitosis for growth and repair, any organ or tissue, usually gets a lot of wear and tear. Think about your skin cells. They're constantly sloughing off. They need to be replaced. If you keep old skin cells around, eventually they're not going to function well. They're not going to be able to produce the proteins and the things that you need. So we want only healthy cells with functionally intact DNA to reproduce. Loss of apoptosis is bad. If we can't get rid of the cells that are old and damaged, they will keep multiplying, and they will multiply in a way they are non-functional cells. So that's the hallmark of the cancer cell, a cell that won't die even though it doesn't function the way it should. And usually the oncogenes kind of accelerate cancer cells, cancer growth, because suppressor genes tend to be diminished. And that's more of a topic for epigenomics, which is coming up soon, but not yet. All right, I'm just going to kind of go through that process again with a little analogy. It's mission apoptosis. All right, so you got your little spy guy. I want you to imagine a cell, a functioning living cell in any of those tissues that undergo mitosis as a sleeper agent. He's been planted there, living a normal life, waiting for instructions. One day that agent gets a transmission from a man whose code name is oncogene. He is the gene that promotes growth. The message says, your mission is to recruit and train 2 new spies who can function exactly the same way you do. We've been compromised. We need more people on the ground. So that's his mission. But first he has to stop at a checkpoint and make sure he has all the clearances and the equipment for the mission. And that's the same thing the cell does. The cell will check and make sure, do we have everything within this cell to reproduce? Do we have all the materials? Are we healthy enough to reproduce? Is all of the genetic information in the right place? Do we have enough oxygen? Do we have enough, um, stuff, ingredients for new cells? So if the spine makes it through that, he has to keep going. He can't stop until it's over. He has to go through that whole spice cycle. He has to complete the mission. And he relies now on intelligence from inside the cell. Okay? If our spy makes a mistake, he gets arrested. If he jeopardizes the mission, he dies. Okay? Does that kind of make sense? If it's more confusing, just skip it. But if it helps you understand it, I kind of like to think in analogies because it's hard for me to picture all this stuff happening in a cell when it's all chemicals and long enzyme names. So when a spy's been on one too many missions, say he starts getting sloppy, now he's not spotting enemies. He doesn't know he's being trailed. He gets taken out of the field. So you can decide whether you want to imagine a gold watch retirement for this cell or a dark alley with, somebody in a silencer. But there it is. It's mission apoptosis. He needs to be retired. He's no longer useful to any more missions. And there's enough new agents to take over for him. So that is that. And we're going to kind of let go here. And the next topic we'll come up with is embryonic development.